My name is Kirill Vinnikov. Today I would like to present you the EXONtools pipeline. Specifically, we would like to show you how we tested the pseudoreference development and hybridization by design for exon capture experiments using our pipeline. And I'm doing this research project with my former PhD advisor, Kathleen Cole from the University of Hawaii at Manoa. So with the rise of next-gen sequences approaches 10 years ago, today we are able to obtain the whole genomes, transcriptomes, or we are able to genotype multiple individuals across their genomes, or we can compare even populations by their genomes. And the greatest interest in such studies is shown by the protein coding regions because they include signals for predicting different phenotypic changes or particular reactions to the environment. So that's why in our studies we put the greatest effort on how to extract those protein coding regions and compare populations using them. In order to get significant results in such comparisons, you need a lot of replicates. That's why it's very important that you sequence multiple individuals for each population or for each experimental stage in order to obtain significant results in comparisons. So it means that you have to sequence a lot, but by extracting only protein coding regions from the whole genome, you save a lot of sequencing effort that you can use to sequence multiple individuals altogether in one sequencing run. For example, in my PhD study, I sequenced more than 100 individuals in one Illumina lane. So how to extract only protein coding regions and then sequence them? First, you have to share your DNA into small pieces, and then using the hybridization baits and magnetic beads, you can extract only protein coding genes from those samples. The only problem is that in order to design these hybridization baits, you need some sort of a reference genome. These kind of references are available only for model organisms today. But for non-model organisms, we propose to use some available transcriptomic data, or you can produce your own transcriptomes, which is not super hard. So by analyzing RNA molecules, you can predict exon sequences and use those as templates for future hybridization blades. So these transcriptome assemblies are, in our study, we call them pseudoreference. And pseudoreference meaning that they do not represent a particular organism, but or a specimen, they belong to multiple organisms, like multiple tissues, multiple individuals, or even multiple species. And then you can use this template to produce hybridization baits that are specific for particular taxa, either general or even family level. So this experimental work includes three main stages. First, you have to do transcriptome sequencing, usually de novo, because if you work with non-model species, you usually don't have transcriptome data. Then you have to assemble and predict some good exons that can be used for bait design. And then you design those baits, you hybridize them to your DNA, like across hundreds of individuals, and then you extract those exons and you can sequence them in one sequencing run. So on each stage, you have a lot of data that should be somehow analyzed using bioinformatics. And that's why we propose the EXONtools pipeline that allows you to analyze all data on each stage and produce the results that are required for population studies or for phylogenomic studies. Currently, EXONtools is under construction because we still haven't published this paper, but soon it will happen. So the first stage of this analysis is that you have to collect raw reads, you have to clean them, and then using different assemblers, you have to produce multiple assemblies, mix them all together across individuals or across species, and then to reassemble them, what we call as a consensus assembly. And then you can call variable sites, predict ORFs, and make further annotation, like functional annotation across all transcripts. And finally, you got the set of annotated transcripts, what we call pseudoreference, that can be used for a hybridization-based design. So when I say consensus assembly, it means that we can take several assemblies from different programs, recombine them all together, and produce some sort of consensus assembly, removing all the redundancy. And you can do it on multiple levels, either assemblers, individuals, or species, and further. So in our test, we use several test species from two genera, freshwater species avals and freshwater species from the genus Stenogobus. So in order to produce high quality assemblies, we first clean all sequencing reads for 5 individuals and use them in 3 different assembly programs that are state-of-the-art today. So these are Spades, Transabyss, and Trinity, and here you can see that they produce different numbers of raw contigs or raw transcripts. And then by using EXONtools consensus assembly, you can reduce the redundancy in those results. But you see that reduction is not uniform across the assemblies. On this slide, you can see how many reads were mapped across produced assemblies, and see that the number of read duplicates is quite high across all assemblies that we produce, which is quite predictable. So when we produced species pseudoreferences or species consensus assembly, those were different because we used different number of individuals within each analysis, but the quality of each assembly was quite similar. Then we took those species assemblies, or we can now call them pseudoreferences, and annotated them using different available annotations in the gene bank from different species. And we see that closely related species provide more than 70% of all annotations, but also 30% were predicted by many other different species, which is a very important result, I think, because usually people are using only one reference genome. Here we comparing how many new annotations were found in a particular assembler program or in particular individual. And this slide shows that combining different individuals or different assemblies is very important for creating the pseudoreference. So when we compare annotations across different assemblers, we found that Transabyss produced the highest number of unique annotations, but also it is interesting that the reduction of those assemblies provide quite similar, make all the results across assemblies quite similar to each other. So the difference is not high across all assemblies. Also interesting that in the initial assemblies, we have a lot of isoforms, and EXONtools pipeline provides you the opportunity to switch the threshold for contigs by their identity. And then you can significantly reduce the number of isoforms in your pseudoreference, which is important. Also, because we mixed a lot of data that is not belong to the same organism, we have a lot of chimeric contigs, and EXONtools provides several approaches how to filter those and make your data look nicer. Interestingly here, the Trinity has a much greater number of contig chimeras than 2 other assembly programs. Finally, we obtain this pseudoreference data. that included many different contigs, and as you remember, many of those were annotated and predicted different contigs. Also had heterozygous sites, and some of them were homozygous, and all the contigs were quite different length. And here you can see also the outlier that is shown here on the slide. This is a mitochondrial DNA that was also included in our analysis and was covered by EXONtools pipeline. So later on, we collected all these contigs from pseudoreference and split them by exons, and use those exons to predict future baits that can be used for exon capture experiment. But before we did this prediction, we also filtered those exons by their length and other sequence qualities like GC content and many other things. And finally, we obtained 130 exons that can be used, 1,000 exons that can be used for bait design. And for our particular experiment, we created almost 20,000 exons and used them for baits. And those exons represented almost 12,000 different genes. So in conclusion, I would like to say here that we present and test EXONtools pipeline that allows us to create specific pseudoreference from RNA-seq data, and you can use these pseudoreference to reconstruct exon capture baits and then do the exon capture experiment. So by using the consensus reassembling approach, we can increase the gene diversity in our pseudoreference. We can increase contig numbers, and we can also improve the contig quality, like lengths. And in our particular experiment, we were able to generate more than 25. We were able to found, and in our pseudoreference, more than 25,000 unique genes. And after filtration and other approaches, we created a custom exon set that was used to design 4K hybridization baits for 20,000 exons that were representing almost 12,000 genes. So thank you very much for your attention, and I would be happy to answer the questions at the QA session. And see you all very soon. And let's do the exon capture. See you. Bye.